material of the host cell. Good, we got that one. And then we talked about malaria caused by a protoctista. Then we said immunity means a body is protected from the invasion of a pathogen. When will I be immune? I will be immune if I have lymphocytes that can produce antibodies. So my body can produce its own antibodies and my body then can fight. Now I want us to continue and see how do my body fight. I can have a first line of defense. The first line of defense can either be a mechanical barrier, which means my skin is a mechanical barrier. Do you understand that? The lining inside my mouth is a mechanical barrier. But with that lining inside my mouth, I also have a chemical barrier. Because in my mouth I have saliva, which is a chemical substance. Nah? And together they form a first line of defense. So if there's a pathogen in the air and my saliva with its antiseptic qualities, you remember that word? Saliva? Antiseptic qualities can kill that pathogen. It is a first line of defense. My eyes is always touching the air. In my eyes, I have a chemical. What chemical will I find in my eyes? What do we call those chemical? Tears. Tears are also antiseptical. They taste salty, don't you think so? And when you want to preserve your food, you add salt. No. So, actually, those tears in the eyes is part of the first line of defense. There's a chemical that can prevent the pathogen to enter. Now let's go into our respiratory tract. And there we have an epithelial layer. And what is on top of those epithelium cells? Hair-like structures, cilia. And they are part of the mechanical barrier. Because what will they do? They will move always and make sure that the pathogens is moved out of the respiratory tract. But there's also mucus, né? And this mucus also helps to trap. So the mucus is part of the chemical barrier. Okay, we can then move into in the nose we have the hairs and the mucus, and in our ears we have those sticky brown substance, and that sticky brown substance is a chemical that helps to kill pathogens. Now, it's not only there to irritate you. Okay, then we go into the stomach, and in the stomach we have acid. Which acid? Can you remember the name of that acid? Hydrochloric acid, which helps with digestion, but it also helps to kill pathogens. Then we can go to the urethra. The flow of urethra, the flow in the urethra helps to prevent bacterial growth. We can go to the vagina, where we have an acidic secretion. It also prevents pathogens to grow there. And the anus has mucous membranes, which will also trap the microorganisms. And of course, there is transport from the anus outwards of the feces. And that is our first and chemical, first line of defense, mechanical, making a barrier, and chemical, which is substances. Okay, you got that. Then, thirdly, we have the white blood cells. Now, the white blood cells can only act when the pathogen is between the cells, or when the pathogen is where? When the pathogen is between the cells, or in the blood, or in the lymph system. Can you understand that? When it's in the blood, when it's between the cells, or when there are pathogens in the lymphatic system. And yes, we know that we have phagocytes, and they are the ones that engulf and digest. And we will watch a video now. Or we have the lymphocytes. What? Tell me. And they will make the antibodies. And there I have a very nice photograph. I just wanted to show you that photograph. It's a photograph. And what is that? Which one? Which one? It's a phagocyte. Can you see it wants to eat? Nah, phagocyte. Okay. I want to show you a video of phago. You can read there. Okay. And there you have this very dramatic. Yeah, you will see it with a microscope. A very good microscope, of course. Now, in your textbook, in your NAMCOL textbook, you will see there is a nice set of pictures that is telling you the steps of phagocytosis. Please make sure it is what we have seen now. Nee, but make sure you know the steps as they occur. Okay, right. And then, of course, we go to, what is the name of the other white blood cell? The lymphocytes. And they are produced in your bone marrow, but then they migrate to your lymph nodes. And they are waiting in the lymph nodes to detect pathogens. But the phagocytes, the moment that they recognize something as foreign, it will be eaten up by phagocytosis. The lymphocytes are very specific because they are making antibodies, and the antibodies must have the same shape as the antigen. So let's make sure, where will I find the antigen? The antigen is on top of the pathogen. Will you remember it like that? Antigen is a protein. It's a receptor, and it is on top of the pathogen. Now, that pathogen with its specific receptors is welcomed in the body. It means your body does not really know, now I have to kill it. But we need the soldiers to look for all these foreign objects And if the lymphocyte recognize foreign antigens, the lymphocyte will make an antibody. The antibody shape needs to fit on the antigen, and the moment when it fits, it will deactivate the pathogen, or it will make the pathogens forming groups. And now it is easy for the phagocyte to destroy it by phagocytosis. Okay, you will see this in the next video. So, what we now discovered was active immunity. Active immunity is when my body, now you must listen carefully, active immunity is when my body can make its own antibodies. Ne? But not only antibodies to destroy the pathogen now, it will also remain or leave a copy behind. And later in the season of the flu or later in your life, when that same pathogen enters, there are memory cells present, and which can immediately react. Therefore, we say active immunity means my own body produce the antibodies. Active means own body. It is long-term protection because when the antigen is identified, the correct specific antibody is produced and it will eliminate it. Remember, all the antibodies that attach are destroyed. They are destroyed the moment when that pathogen is destroyed. So, they need to stay a copy behind. The name of the copy is the memory cells. Nee, sorry, ek het hier aangeskryf. Is the memory cell, which is a lymphocyte that specially stays behind, not become part of the war against the pathogen. If the pathogen enters again, the memory cell are quickly ready and they can destroy before you are even aware of the disease. You don't have any symptoms because your body can fight it off. Do you understand that? That is your active immunity. But we can also have passive immunity. I repeat, because this is something that can confuse us. Active immunity means my body fight the war itself. I'm actively involved. Passive immunity means I can get help. I can acquire antibodies from another person. Okay? I can even acquire antibodies from another mammal. I will explain that later. Now, I acquire antibodies. So what do we know what happens to the antibodies? They are destroyed during the war. Therefore, it is short term. My body does not have the recipe to make the antibodies. You agree? Therefore, it's short term. I get the antibodies from something else. The antibodies are fighting the war, but they are destroyed during the war. So it's a short-term protection, short-term immunity, because there's no memory. Okay. Do you understand? Active, own body, own fight, own antibodies. Passive is the antibodies came from another human or another organism. Okay. And here I try to make a summary of passive and active. Person does not make memory cells or antibodies. The antibodies only are transferred, so it is not long-term, it is short-term. Examples. I can have a natural passive immunity because I am a baby and I get the antibodies from my mother through breastfeeding. When the fetus are in the uterus, there's also antibodies moving from the mother into the fetus. That is natural. But is it active or passive? Passive because it came from another. It's not own made. Nee. Artificial is when I have a vaccine, when I'm vaccinated. When I'm vaccinated, I receive the pathogen. I receive the pathogen. My body, sorry, I am lying. The rabies shot is something else. The rabies shot, I get antibodies that is produced in the body of a horse. They take out that of the antibodies, and that liquid they call serum. Serum. S-E-R-U-M. Now I am injected with antibodies that was produced by a horse. And those antibodies help to fight me. The horse will not die. The horse will only has a large body and the horse can produce it. Okay, that is passive, artificial. Artificial means it was deliberately made. Nah, natural, it's a natural process. The mother made it for the baby. But artificial means it is a deliberate process happening outside in a laboratory. Active, I think we do understand active. The body makes its own antibodies, long-term immunity because memory cells are made. Natural, the moment when your body is fighting a disease, it is active immunity on the natural way, but it can also be the artificial way. Artificial way means you are injected with a pathogen, né? But a weakened one or a dead one or only a part of a pathogen. But my body has time for those small amount of pathogens or weakened or inactivated, and now my body makes its own antibodies. Therefore, it is active. Okay, right. Let's get to our next video. I quickly want to show this video first before we talk further. Did you see this one, perhaps?
In 1796, the scientist Edward Jenner injected material from a cowpox virus into an 8-year-old boy, with a hunch that this would provide the protection needed to save people from deadly outbreaks of the related smallpox virus. It was a success. The 8-year-old was inoculated against the disease, and this became the first ever vaccine. But why did it work? To understand how vaccines function, we need to know how the immune system defends us against contagious diseases in the first place. When foreign microbes invade us, the immune system triggers a series of responses in an attempt to identify and remove them from our bodies. The signs that this immune response is working are the coughing, sneezing, inflammation, and fever we experience, which work to trap, deter, and rid the body of threatening things like bacteria. These innate immune responses also trigger our second line of defense, called adaptive immunity. Special cells called B cells and T cells are recruited to fight microbes and also record information about them, creating a memory of what the invaders look like and how best to fight them. This know-how becomes handy if the same pathogen invades the body again. But despite this smart response, there's still a risk involved. The body takes time to learn how to respond to pathogens and to build up these defenses, And even then, if a body is too weak or young to fight back when it's invaded, it might face very serious risk if the pathogen is particularly severe. But what if we could prepare the body's immune response, readying it before someone even got ill? This is where vaccines come in. Using the same principles that the body uses to defend itself, scientists use vaccines to trigger the body's adaptive immune system without exposing humans to the full-strength disease. This has resulted in many vaccines, which each work uniquely, separated into many different types. First, we have live attenuated vaccines. These are made of the pathogen itself, but a much weaker and tamer version. Next, we have inactive vaccines, in which the pathogens have been killed. The weakening and inactivation in both types of vaccine ensures that pathogens don't develop into the full-blown disease. But just like a disease, they trigger an immune response, teaching the body to recognize an attack by making a profile of pathogens in preparation. The downside is that live attenuated vaccines can be difficult to make, and because they're live and quite powerful, people with weaker immune systems can't have them, while inactive vaccines don't create long-lasting immunity. Another type, the subunit vaccine, is only made from one part of the pathogen, called an antigen, the ingredient that actually triggers the immune response. By even further isolating specific components of antigens, like proteins or polysaccharides, these vaccines can prompt specific responses. Scientists are now building a whole new range of vaccines called DNA vaccines. For this variety, they isolate the very genes that make the specific antigens the body needs to trigger its immune response to specific pathogens. When injected into the human body, those genes instruct cells in the body to make the antigens. This causes a stronger immune response and prepares the body for any future threats. And because the vaccine only includes specific genetic material, it doesn't contain any other ingredients from the rest of the pathogen that could develop into the disease and harm the patient. If these vaccines become a success, we might be able to build more effective treatments for invasive pathogens in years to come. Just like Edward Jenner's amazing discovery spurred on modern medicine all those decades ago, continuing the development of vaccines might even allow us to treat diseases like HIV, malaria, or Ebola one day.
Okay, right. Ekskuus, ek het nou ergens sorry, I missed one of ek het nou ergens hier. Nee. I missed one video that I want to show you. Okay, sorry, I quickly want to show you this one. Yes, this is a nice one because it's going to explain a lot of what you want to understand. Ach nee. Okay.
A pathogen is a disease-causing organism, and there's a few examples of them shown here on the screen.
Nee, julle kan nie. Okay, you can pack up and you can leave the class and you
A pathogen is a disease-causing organism, and there's a few examples of them shown here on the screen. Your body's fairly well equipped to try and deal with it if a pathogen does get in there. We're going to discuss this in the context of some of the blood cells that are involved. Now, you obviously know about red blood cells. They are there for transporting oxygen. There are also lymphocytes and phagocytes. Lymphocytes and phagocytes are both types of white blood cells. If we have a look at what they really look like here in this microscope image of the blood, we can see first of all this red blood cell. I've highlighted one of them here, but that's the one where you can see hundreds of them on the screen. The lymphocyte there, we can tell that that's the lymphocyte because it's got that really large nucleus. And the phagocyte down here, we can distinguish that from the lymphocyte because of the unusual shape of its nucleus.